ultimately, that experience really solidified that my personal space is more in the biotech side of things, in particular in the early stage biotech, where innovation happens very nimbly, very fast. You can also take a few more risks along the way.
Hello and welcome back to Innovation Insights. We are very excited today to be at LabCentral here in Cambridge, Massachusetts, in the greater Boston area. For those of you that don't know LabCentral, it is one of the largest shared wet lab facilities in the US. Startups here have raised over $20 billion in funding by all the residents that are here in the last 11 years since LabCentral was created. Over 16,000 patients have been enrolled in clinical trials from innovations coming from the startups in LabCentral, and it has created over 7,000 jobs. I'm joined today by Dr. Johanna Kaufmann. Thank you very much for being here. She's the Chief Scientific Officer at Bio. It's an early stage startup that is focusing on oncology, developing innovative T-cell engagers for solid tumors. So Johanna, thank you very much for being here. We're going to start soon.
Yeah, thank you for having me, Victor.
All right, so can you tell us a little bit about what was your journey to become what you are today, the Chief Scientific Officer of this very cool startup?
Yeah, thank you. So I'm a cancer biologist by training with a PhD from German Cancer Research Center, which is essentially the German version of the NCI.
All right.
And I was able to combine two scientific passions during my PhD project. On the one hand, there were viruses. I just think it's fascinating that these tiny little things that are technically not even alive can wreak so much havoc in patients. And on the other hand, there was cancer research. I had the opportunity to spend some time abroad during my high school times and learned the void patients leave behind when they pass away from cancer in their families and their communities. So I wanted to do something about that disease area. And I found a way to combine those two scientific areas in the form of oncolytic viruses, which are viruses that are engineered in a certain way that they can specifically infect cancer cells, replicate in them, destroy them, leaving healthy cells alone. At the time I started my PhD, we thought that the immune system was actually bad for this kind of therapy because we thought that the immune system would recognize the virus as foreign and eliminate the virus, which in this case is a drug, not a pathogenic agent. However, throughout my PhD, we as a field learned that the immune system is actually a friend, not a foe, because all that debris that the virus leaves behind of the tumor cells is actually stimulating the immune system and re-educating it to attack the cancer in the patient. So as oncolytic viruses turned from something like a gene therapy to an immunotherapy, I became an immuno-oncologist.
There you go. Okay.
And then I spent about the last 12 years or so of my career in industry in a variety of different companies and roles that span the drug discovery or drug development spectrum from discovery to mid-stage clinical trials, generally with an immuno-oncology focus or adjacent areas in infectious disease.
Okay, so you were completely focused on your PhD, doing this research, which started about how to take evil to do good.
Exactly.
And then
Trojan horses is what we called them, yes.
All right, and then how evil and good can go together. Basically, you were going on that direction, which is very philosophical, but also scientific. And then you decided to go and work for industry. Can you explain a little bit why you are taking that choice? Because I want to understand how do you go from the academia, then to industry, then to the innovation startup world, right? So there is a journey here.
Yes. Great question. My objective for my career was to make a difference for patients suffering from cancer. And during my PhD, as well as during my postdoc, while I was working at sort of translational research hospital kind of institutions, I still felt there is not enough impact, and I didn't see the translation of the science we were working on to the patient directly. And at the time I thought, and by now know, that that by working in industry in a variety of forms, that translation is much closer to what a scientist at a bench can observe and see. And I was fortunate enough that also my first company that I worked for was a clinical stage company at the time I joined.
I see.
And while my research project was in preclinical science, working on the next generation of an asset that was already in clinical trials, my team also supported directly assays that helped us understand what our drug actually did to patients. And so I felt immediately connected to that translational aspect that got me into this career in the first place.
Okay, so let me then follow your journey. You are going into the clinical trial. So through the academic discovery, you are seeing, okay, this is fantastic, but I want to see what is the impact.
Yeah.
And then you go directly to see the impact with the clinical trial part, and you're working with this company, which goes from the preclinical trial of the more academic one to the actual real clinical trial, and you start seeing, okay, this can have an impact. What is next? Because then you continue your evolution. What is your next step in your career?
Yeah, I think the next sort of really important part was working for a company that got acquired by Big Pharma. So I spent some time at GlaxoSmithKline as a member of the senior leadership team of the Immuno-Oncology Combinations Research Unit there, which I joined through the $5 billion acquisition of Tesaro. And that was actually an amazing learning opportunity to see how pharma operates, how they think about the drug development process. And I was fortunate enough to work with amazing leaders and scientists there. And you see the scale drug development can have, but also has to have to actually proceed from a new idea of a drug through clinical development into the commercial application of that. So it ultimately actually reaches patients. It was a fantastic learning opportunity for me, but I also saw some of the, let's say, bureaucracies and hurdles that come with operating a company of that size. And so ultimately that experience really solidified that my personal space is more in the biotech side of things, in particular in the early stage biotech, where innovation happens very nimbly, very fast. You can also take a few more risks along the way in order to really push the boundaries on the science to make a difference for patients. After leaving GSK, I joined a biotech company that was 20 people large at that time, but my role was to found an oncology department here in the Cambridge area. So it was in a way building a startup within the confines of a biotech. And the lessons that I learned from that process, I'm now taking to Deck Bio, where we are 3 people and really are a true startup in the real sense.
Love it. Because at the end, I mean, there was for me a very big lesson from during the COVID times. I think that was a great lesson, because you see there are 2 models where you can do this very rapid innovation, scientific discovery, and apply it. That was the lesson of COVID. We can do that fast with the right mechanisms. But for me, it was very interesting to see the 2 successful ones. One was here in the US, was Moderna, was in this ecosystem. It was in the greater Boston area, which is ready to do that. It's ready to take a startup, find all the pieces around it, and that was Bob Langer. I mean, of course, he has the ecosystem, and he knows who could be the right CEO to kind of scale it. But that was more of the complete startup route with VC and going that way. And then you have the German one. It's very interesting because that's Biotech, and Biotech said, yes, we can develop the solution, and it's parallel and different, but we are going to partner with a big pharma to actually do the scale. And actually, you see the impact, they were more successful in terms of reaching out to people than Moderna. In terms of business, I don't know. Maybe Moderna was more successful, but maybe I don't know. But in terms of impact, you can see the difference. So very interesting what you were saying, because at the end, Big Pharma with all this bureaucracy is the one that is going to scale up. And then the innovation is coming from the startup. So that combination, I think, is amazing, and you have both experiences. And I think that brings a completely different mindset.
I think the R&D continuum stands on 3 pillars, right? There is the basic discoveries of major biology and pathways. Most often that happens in academia. There is then the translation of these kinds of tools or insights into potential drugs, and that's where biotech generally shines. And then there's ultimately the clinical and commercial development, which pharma really knows well how to do. Ultimately, it takes all three of these pillars to bring a drug to patients. This drug development process is very long, depending on which publication you read, 8 years, 10 years, depending also on the disease area. So it takes the combination of these players in the ecosystem to make this happen. And it is places like LabCentral or the CIC that bring these players of the ecosystem together to accelerate the connection points between the different pillars.
Yeah, and what I love is you have this complete mapping and experiences to understand how to engage and when to engage with the others. How did you find your co-founders? You mentioned 3 people, but
I technically didn't found DeckBio. I'm part of the founding team. It was founded in September 2023. It was originally a one-man show, to be honest. Our CEO and founder, Jack Silberstein, brought together his experience from the protein engineering side, together with what he saw as clinical gaps when he was a clinical trial coordinator, enrolling patients into clinical trials, amalgamated these 2 experiences into what is now DeckBio, got accepted to build his company here at LabCentral, and then brought on a head of protein engineering about half a year later, and I joined another half year later or so to bring the operator qualities and the immunology translational side of things to the table. And we need all of these expertises to really advance our lead asset, DBX01.
All right. So what was exciting about you about joining this project? I mean, you're going from different kind of experiences. You've been with the big pharma, you've been with the other, and then suddenly you said, yes, I'm going to join this 2 people so.
Yes. It was actually a combination of things, and maybe my pharma experience influenced how I thought about this particular opportunity. What we're doing at DeckBio is not just combining a modality that we believe has potential for significant impact for patients that needs to be taken to the next level, and that's where our scientific innovations come into play. But there was also a tools component. How do we make this drug the most specific it can be, and therefore we are avoiding potential off-target toxicities that patients suffer from. And then the final kicker for me was actually the IP-generating technology that underlies how we build our T-cell engagers, which improves the developability or manufacturability of our molecules. And I thought that was actually quite important to put something that people might think about as an afterthought, as in like pharma can figure out how to make this, and we put this aspiration of we want to make this a lot easier to manufacture than competitor molecules have been, and put that at the very front of our design criteria.
I love
it. That I really thought was important for me to make sure that I can see the future of our assets and how we actually bring this to patients, even though, of course, our work in the first couple of years has been on the very early discovery side of things.
No, I love it. And that's one of the advices for especially biotech, life science companies that I see the most from programs like StarX in Stanford, and they say like, okay, think from the beginning how this is going to be commercializable.
Exactly.
Think from the beginning what is needed from the clinical trials approval and FDA approval that you're going to need. Don't wait until that happens because it will be too late, and you're doing it from the beginning. And I think bringing your experience with big pharma and understanding what they will need and how easy it will make it makes you special in that sense, and I think that's unique. But tell me more about what is unique about your company right now. What are you doing that is unique against the market? Because we talk a little bit in general about the area, but Explain a little bit more. What is specific about it?
Yes. Let me take a step back first and say T-cell engagers are molecules that essentially bridge two cell types together, with one side, in our case, that's a cytotoxic T-cell, with the other side, in our case, that is a cancer cell. And then the idea is that when these two cells are being brought together, you mediate cancer cell killing through that T cell.
Okay.
This is very well established in hematological malignancies with multiple FDA-approved drugs that drive over $3.5 billion in annual revenue. However, translating that into solid tumors, which are actually 90 to 95% of all tumors that patients suffer from, has been lagging behind. So if you now think about how you would naturally engage a cancer cell specifically, you think about cell surface proteins, because obviously a biologic needs to recognize this cancer cell somehow. The problem with these cell surface proteins, however, often is that there is residual expression on healthy cells.
I see. Yeah.
And while that may be okay, that differential of expression levels in cancer versus healthy cells for a modality like antibody drug conjugates or traditional monoclonal antibodies like those that target HER2, for a highly potent modality like T-cell engagers, this is a real issue. And we have seen examples of healthy tissue expression leading to actually fatalities with these kinds of modalities. So obviously that was a no-go for us. And that is why we are working on a different target class that is much more tumor exclusive, and those are peptide MHC complexes. So these are actually generated as part of natural immune surveillance mechanisms of the body, where every protein that's made in a cell gets essentially sampled in bits and pieces and gets displayed on the cell surface. So the first piece is we're taking tumor exclusive targets. And there is clinical precedence for this type of approach. Now, the problem with this is that often there is very low expression levels of these peptide MHC complexes, and that has really hampered the widespread use of this approach, right? So DEC-Bio's innovation really tackles this expression level problem by actually identifying signatures of multiple tumor-exclusive targets that generate peptide MHC complexes that can be recognized by a single drug. So this multi-targeting approach addresses concerns about expression levels. Therefore, more patients will meet expression levels required for clinical response. It addresses questions around heterogeneity, either between patients or within cells in a tumor, and it gets ahead of certain types of resistance mechanisms in the same process.
All right. So now let me see if I understood everything. You are basically doing very precise assessments of tumor cells. And what you are avoiding is that they don't go to whatever is not a tumor cell.
Correct.
So that's importance number one.
Yes, exactly.
And you are doing that by also providing a way to do that is much broader than what existed before. Because otherwise it was too limited and could not address all the different kind of tumors or all the different kind of cells, or you have problems with people being a little bit different and this has been a little bit different, and then you're making it more precise. So in a sense, you are scaling something, giving more versatility to a tool that is very interesting in targeting in a very precise way.
Versatility is a perfect word, because it might be that you might be expressing targets 1 and 2, and I might be expressing target 2 and 4 of our target signature for our lead asset DBX01, which targets 4 different target proteins, but we both will be eligible for this therapy. And that is actually an important piece when people start thinking about sort of the market and the commercialization for a drug like this. While precision medicine is extremely valuable in identifying the patients that have likelihood to respond to a particular drug, and you avoid treating patients with things that are not expected to work. At the same time, that means you're slicing and dicing your patient population, and certainly the commercial folks don't appreciate that once you get into too small slices of the pizza pie. And so broadening that back up and opening larger patient populations is really what's the goal behind DEC-Bio, not just from a definition within a tumor type, but by selecting certain types of targets that are expressed in major solid tumor indications like lung cancer and others along the aerodigestive tract. We're really essentially amplifying the potential patient population eligible for this type of therapy by an order of magnitude in comparison to competitors.
Yeah, no, I love that, because you're addressing the market kind of failure, which is because you really want to have this personalized or precise kind of therapy, but it's so difficult in the market. I mean, the technology exists, but the market is not ready. So you are going to say, okay, I'll solve the market issue. And then the second part, I guess, you can address also a population that was underserved before because the heterogeneity problem, right? I mean, you probably develop the drugs for a very kind of target population tends to be, which is the one that the market can address more, and they are developed primarily in countries like the US or in Europe or in Japan. And then what other populations that can be a little bit different or heterogenic, right? So in that sense, you are broadening that. So
we are broadening that in that sense. I'm not claiming that our approach is tailored towards developing countries of that kind of underserved communities, but it is broadening the patient population. I think another piece to pay attention to here for peptide MHC targeted modalities specifically is that these are tailored towards individuals of a particular HLA type, right? And for our first program does attack peptide MHC complexes presented in the context of A0201, which is the most prevalent HLA type in the US and Europe. However, for future programs, we will be thinking about translating that approach in the context of additional HLA types. In particular, for example, HLA 2402, which is prevalent in about 70% of the Japanese population, for example. And that is another way how we're broadening the patient population by covering diverse genetic backgrounds through different HLA types.
No, I love it, because at the end it's just the approach that then it's scalable, and then you're providing the tools to do that, and it's very clever to address the market kind of failure there, which I think your experience is also bringing. So I think it's very unique approach, and everybody is looking forward to this personalized kind of therapies or personalized medicine, and I think this gives a good segue in how this is possible going forward. Now, a question for you. This is fantastic. It's one of the frontier areas that we are working globally in the world, in the ecosystem. How do you see this evolving? What is exciting for you that there are the new things that are coming here, either they are complementary to what you're doing, or they say this is very exciting in the field?
Yeah, I think there are two tensions, I would say, here. Personally, I really appreciate the precision medicine approach. In order to really make this scalable, we need better technologies to identify patients that are eligible for a certain type of therapy. So the companion diagnostic development that has to go in parallel with such a precision medicine is hopefully something that we can accelerate and parallelize also from a regulatory perspective in the future. I think the tensions on the pharma side and on the investor side who need return on their investment quickly is what's the scale and the size of the market population. And I'm curious to see how that plays out over the next few years in the investor community, but also in pharma acceptance.
How do you see the market where you are operating in 5 years from now? If you say like, okay, if everything goes well, and you do everything right, and the market kind of responds to it, and the bio say like, yes, we nail it, we have all the success that we dream about, and then the kind of the market evolves with that, but also the scientific discovery around you. How is the world changing 5 years from now or 10 years from now when you finish the clinical trials and everything is commercialized?
I think we will have proven that multipeptide MHC targeting as an approach is valuable, and that our technologies that we have developed as platform technologies can really generate ultra-specific binders that are highly developable. Once you have that established and there's proof of concept for that, there are so many other opportunities that DeckBio can follow along. We can go after, obviously, different target signatures. We can even go outside of oncology into the inflammation autoimmunity space. As long as you can identify disease-specific peptide MHC complexes, we can then generate a drug against that. That's the vision once we prove out our technologies are successful. So that obviously opens a whole different scale of patient populations that can be addressed with these types of targets, because obviously the autoimmunity commercial space is a lot larger than oncology, and is also obviously currently very popular with investors and pharma companies.
Okay, so you are focusing right now in the cancer treatment. Autoimmunology, autoimmunology is the next kind of field. Any other field that you say, like, okay, this is the other one or two that we have in mind too, or how do you see that?
Yeah, I think those are the attainable ones at this point in time, where there is some proof of concept. I think something that would be interesting to me within that space is diabetes, for example. There is a pre-diabetic state where there are publications where certain peptide MHC complexes already indicate that someone is on the way towards a diabetic state, and interception and early intervention might be a way to enter that space. And also in the oncology field, interception and intervention is an up-and-coming area that I think we should be thinking about more, so we don't have to go into clinical trials in late-stage cancer patients
that would be much better.
In particular for immuno-oncology might not be the perfect patient population to prove out a mechanism of action.
But I love you mentioned diabetes, because it's completely different than the other two, and it's bringing kind of a different dimension. So it's opening the field for many other kind of improvements about what we are saying. And diabetes is something that it's very widespread, and it will change also the life of a wide set of population. So it's not only specific like this target medicine, but also like a broader kind of set. Love it. I want to ask you, after all this journey and after what you have seen so far and your experience, a little bit of a word of wisdom and advice for someone that is starting in their career and it's in the PhD right now and thinking, okay, I really want to have more impact, or I want to do more innovations. What would you tell them? So imagine, what advice would you give to the Joe that was there when you finished the PhD, and someone came to you and coming from the future, I'm telling you, this is the wisdom for you.
I would say, don't forget the T might be the tagline. If you think about an academic career is where most of us start, at least from an educational perspective, we are trained to go very deep into specific scientific areas. So the downwards part of the T, we're supposed to go very deep and become experts in that field. And what that ultimately makes you hesitate is to go to adjacent sides and bring diverse thoughts, diverse applications of the skills that you learned or the scientific tools that you have learned, and apply them in a novel way. So I feel like we shouldn't forget about the top part of the T, which is the breadth of experience. And when an opportunity arises that seems adjacent and not actually perfectly right, but if it's a good opportunity, take it and learn something. You will always learn something, even if it's just a failed opportunity, or that company ultimately doesn't make it, or that program doesn't make it. But it is breadth of thought and breadth of experience that really makes a difference ultimately to arrive at skills and a vision actually for what you want to do with your career ultimately. So it takes both parts. You do need to have deep scientific expertise in a certain area, but you also need the broad scope, and don't be afraid to work on that broad part.
All right, don't forget about the T. That's
it.
All right, so we are coming to the end of the interview, and we always ask a final question is about how to follow you. If the audience is interested in following you, how should they do it?
Yep, the easiest way is just to follow me on LinkedIn.
All right, fantastic, and we will post the LinkedIn for you guys in the video. Thank you so much. That was wonderful. Thank you, Johanna. It was a wonderful conversation.
Thank you. It was great to be here.
All right, thank you.